and we are going to enjoy more even intriguing plant response and behavior today. So you can start, Yasmine.
Thank you, Well. Good morning to everybody, and welcome to the graveyard shift, as Malcolm put it. Thanks for joining. Okay, so I think we all agree that plants are the best. Maybe I want to just show you kind of my point of view of what I find really intriguing about plants in a very kind of zoomed out way. So here you have this beautiful time-lapse BBC Planet Earth, and you can imagine David Attenborough saying something perfect about it. And the idea here is that you can see this climbing plant that's doing a complex navigational task when you think about it. It's searching for light, it's searching for new support, attaching or not attaching, right? Something interesting is going on here. And this is just one example out of a million examples of complex tasks that plants perform all the time in continuously changing environments, and while their own morphology changes as well due to growth, right, which is different from animal systems. It's significantly more complicated in that sense. Yet, if I now chop this climbing plant, I will see that there is no brain, no neurons, just a bunch of cells that are glued together with only transport of water and signaling molecules. So this is quite remarkable when you think about it from this point of view. And then we can say that basically plants are active physical systems that are able to sense the environment, compute stuff, and then move accordingly. And all of these processes are completely decentralized, right? Because there is no central brain or computer telling it what to do. So somehow the plant is able to orchestrate all the single cells to do something together. And this is what we call in physics an emergent property. And we see it in many systems, such as collective behavior of birds, let's say, right? It's a similar question. But I think that plants take it to a completely different level in terms of just how remarkable they are. And these are the sort of questions that we're generally interested in the lab. So how do we study these computational or behavioral questions? We focus on plant tropisms. So whilst, plant tropisms are super interesting per se, just understanding how they work. But I use them as a kind of a gateway or a window in order to ask something which is a bit higher level in terms of computation and behavior and so on. So what's great about tropisms, to ask these questions, is they're a very experimentally tractable input-output system. I know what the stimuli are, and I can give complex stimuli that change over time, like here, the light source was switched, or there the gravitational, the inclination was changed, right? So I can control the input, and I see a very clear output which I can follow. And now I can basically try to reverse engineer. If I know what I plugged in, and I know what came out, maybe I can try to understand what's going on in this black box inside the plant. And what's also very useful about tropisms is that this is the only equation I'm going to show here, don't worry. They're also theoretically tractable, right? We have a nice model that's based on a model that was originally developed by Bruno Muglia and company, which is it's already appeared in some of the talks here, and it basically connects. So the change kappa is a curvature, so this is a change of the shape, right? If there's derivative in time, it means that you're moving. And these are the signals. This is I is a light signal, and G is the direction of gravity. And I simplified some of the equation just to give the feeling that there's an input and there's an output. And it describes tropisms in the case of constant stimuli really, really well. So this is kind of the machinery, let's say, that we use in the lab, the approach. And I think that what I'm going to try to talk the time that I have today is to give a few examples about how plants integrate sensory information over space, right, when you have multiple stimuli, over time, and if I have time, also how they integrate mechanical properties together with these active processes. So let's start with the first notion. This is Ronnie's poster. It's a bit too late to promote it, but so the idea is that usually in the lab, when we study phototropism, we study it with one light source. But in reality, this is kind of what a plant experiences. And you have lights coming from different directions through the canopy, reflected off other plants and whatnot. So it's something very complex. So it's clear that plants need to be able to somehow, first of all, represent this information, right? The directionality of this information. They need to be able to encode it or represent it somehow, and then integrate it somehow in a smart way. So how do they do that? So this is the equation from before, where I only have the light term on the right, which is in green. And I'll just remind you that although Yoel showed that for gravitropism, your tropic response does not depend on the magnitude of the acceleration, just the inclination. In light, it's different. We know that your response depends on the intensity of light. And this is this nu, okay? It tells you it's a signal transduction function, if you want. Okay, so it tells you how strongly you respond to different intensities of light. And so now this was the original equation with just one light source. The question is, if I have multiple light sources, how will this look like? Okay, I mean, for me, where my research is driven by theory, I first ask, how will this look like in theory? And then I go doing the experiment, and then we can understand how the model is correctly described. So the idea is the following. You have a plant, you have two light sources, I1 and I2. Light is defined as a vector. Vector means that you have a magnitude, so the intensity of light, and the direction. So now we know that the total light that the plant is supposed to feel, right? If I were to measure the light here, it would be a sum of these two vectors, which is this big arrow in the middle. And now we can ask, okay, how does the plant respond to this, right? Because there are different options that we can think of. Does it transduce just the big arrow and respond to that, as if it were just one light source? Does it transduce each of these separately and only then sum it, right? Or maybe it's something completely different, I don't know. So there are various options. And to answer this, we decided to look at a simple system. So this is the something in between, right? There's the full complex real natural system, which is very, very complicated. There is one light source, and there's something in between. Let's add another light source. So it's very controlled and simple, but it allows us to ask these questions. So you have a sunflower seedling, and you have light from the left, light from the right, with different light intensities. And now we follow the trajectories of the seedlings. And here you have basically tip angle is a function of time. And the red colors are the ones where the left light is stronger. The blue color is where the right light is stronger. And as you would expect, the bigger differences are the ones which are farther away from zero. And if the differences are small, you don't move a lot. But the question is to quantify this. What exactly is, how does the difference between left and right set what will be your final angle? And so we plug this into two different types of models, which are super ugly, and I'm not going to show it to you here. But after trying the two different ways, whether you first transduce and then sum, or first sum the light and then transduce it, it turns out that the first option works really well. So here you see basically it's an angle that's predicted by the model. So if I give the model intensity 1, intensity 2, tell me what should be the final angle compared to the measured angle. And you see that there's a straight line. So it means that there is a good agreement between the two. Blue dots are for unilateral lighting. Red dots are for bilateral lighting. And I'm showing this because we also take into account the transmission of light. So unilateral lighting is actually, you can think of it as bilateral lighting because the plant senses light on either side. So it's important to have them on the same line. And what we found through this model at the end of the day is that it seems that the plant indeed first transduces the signal and only then adds it. So we could think of it that maybe this is somehow, it represents these vectors in space as maybe gradients in oxen, where you then sum up different gradients and you get the final direction. But this is, of course, something for the future. Another funny prediction that you get from this is that if you put now a plant with two identical strong light sources on the right and on the left, and then a third weaker light source in front, if it's vectors, then the two strong light sources cancel out, and then the plant is supposed to go to the weaker light source, which is a bit weird, right? It doesn't make sense physiologically. We tried this, and this is indeed what you get. So this is a top view of a sunflower, and the red trajectory shows you start from red at time 0, and you go to the weaker light source, which is here. And here you see, of course, the statistics. So it all makes sense in that sense. Okay, now I want to shift gears, and we talked about spatial representation of stimuli. Now I want to see what happens in time, because obviously, again, if we look at what occurs within, if I'm a plant, then this is my time scale, right? This is how I see the world. So there are a lot of fluctuations, whether it's flickering of light, which is much faster than this due to wind or whatever, whether it's the time of day, whether it's just plants moving around or an animal moving around and so on. So it's clear that it doesn't make sense for the plant to respond to every small, tiny fluctuation in light, right? It would make sense that it should be able to somehow identify which direction on average you have most light and grow in that direction. And this is a work that was done by Mathieu Rivière, a very talented postdoc in our group who is now working with you all. Okay, so how do we go about this? There are very old experiments from 1908, where basically both in phototropism and gravitropism, roots and shoots, it's all over the place. It's a recurring theme. Basically, all of these experiments show that plants do not respond, the tropisms do not respond instantaneously to stimuli, but rather to some kind of history of stimuli. So one example of a class of these types of experiments is the following. Let's say on the left, I have one type of stimulus. I turn on the light, as you can see in this picture, for, I don't know, 1 minute. And then I measure the maximal angle. And that's how I represent my response. Now I have a new experiment. I turn on the light for 2 minutes, consecutive minutes. I look at the response and I will have a bigger response, right? The angle will be bigger. Okay, now we do something new. We do 1 minute and then we wait for some time and then another minute. So we separate now these 2 minutes. And it turns out that the plant responds exactly the same. It doesn't see the difference between the consecutive 2 minutes versus 1 minute on, 1 minute off, 1 minute on. Of course, if I wait long enough now, like the first 1 minute was yesterday, of course I will have forgotten it and I will respond only to the second one. So this means that there is a window of time over which I sum or integrate or do something of the kind and respond to that, to this average. So we described this, we wanted to quantify this in some way. Okay, so I lied, there's another equation. In order to describe this, we resorted to something that's called response theory. This is something that is used a lot in engineering and in physics and in other fields. And the basic idea is it works well with any signal transducer. So it's very relevant for tropisms in that sense. You have, for example, this radio. You have an input, which is the electric signal. You have an output, which is sound waves. Now, if I had my screwdriver, I can open up the radio, see all the electricity wiring. And if I remembered my courses in electricity, which I don't, but I could write all the equations, and then I could tell you exactly for a given electrical signal, what is the expected output. But I forgot my screwdriver. I can't open the box, okay? And this is equivalent to not knowing what is the signaling mechanism for hydrotropism or whatever. And then I can do a mathematical trick. So, and this is response theory. The idea is that I can represent these equations with a function, with just one function. So if x is the input, y is the output. If I have this response function or memory kernel, or however, there are many different names that you can call it, this mu, this tells me basically I take x, I multiply it by mu. This tells me how to weigh different parts of the history of x. And then I integrate. To integrate just means to sum over everything. Okay, so this is the averaging. So I'll give you just an example so that you can get a feeling for it. So let's say this is the input signal we talked about before, 2 light signals. And let's say this is the form of a memory function, where I know it goes to 0 at some point, because at some point I know that I forget stuff. So now if I multiply these two, this is what you get. You get a big weight, or you take into consideration more seriously the recent stimulus at T1, right? It's multiplied by a big number, but the older one is multiplied by a small number. And if this second stimulus were here, it would be multiplied by 0. So that's the forgetting. And then, so this is the weight, and now I just sum over this. This is the integral, and I respond to that. Okay, so through simulations we showed that this indeed reproduces qualitatively the experiments that I told you before, but we wanted to become more. Thanks. I just like to walk around. Wanted to be more quantitative. So basically, what I'm saying here is that this memory kernel is really important because its shape represents basically the underlying equations, the underlying circuitry. So if I know something about the real shape, and not just like here where I guessed it, it would tell me something about the biology. So that was Mathieu's next challenge. We did these intermittent inclination experiments. You have wheat colloquials, and you incline it for a certain amount of time, this tau s, and then you incline it back to 90 degrees. So we did it from the shortest inclinations of 5 or 6 minutes, where you can still see a good response, and all the way to a permanent stimulus. Here you see 2 examples. The red line is the tip angle for a permanent response, and the blue line is the response to a short 6-minute inclination. And you see that it's significantly different, right? It's going also in the opposite direction. There's some funny stuff going on here. And from these equations, from these trajectories, we can solve the equation for the memory function and find how this memory function is supposed to look like. And this is its shape. Okay, we have a positive peak and a negative peak, and then it goes to 0. So this is very similar to what we guessed in terms of the positive part and going to 0. But what we didn't see coming from a million miles is this negative peak. And we were very surprised by that. And it's not clear what does it mean? What does it mean to multiply by a negative number? Let's do a thought experiment. If this is, the blue line is this, it's a schematic for the memory function. And like I showed in the example before, let's think about what would be the outcome. So as a control, right, or something to compare to, we compare to just the response of one stimulus. That's on the left. Now, if I have 2 stimuli and they're close together, they're both within the positive peak. So like before, I just sum them basically. They both get a positive weight and I sum them. Fine. If I wait a long time between the 2 stimuli, then the older stimulus would get 0, so I forget it. And then I respond to that case as if it were just one stimulus. Okay. But the interesting part is in the sweet spot. What happens when the timing between the 2 stimuli is such that the recent one is in the positive peak, but the older stimulus is in the negative peak? So mathematically speaking, this means that I'm supposed to give a negative weight to the older one. So what does this mean? If now I integrate over this, right, I'm supposed to sum over this, it means that I'm subtracting the 2 stimuli, which is curious, right? So effectively, in terms of the experiment, this means that for two close stimuli, I expect the response to the second stimulus to be stronger than if it were just one stimulus. If they're in the sweet spot, let's call it, then I'll expect the response to the second stimulus to be weaker than if it were just one, right? A smaller angle. And if I wait long enough between the two, then it will be the same response as if it were just one. So we now ran these experiments. So here, every point, the x-axis is the waiting time between the two 6-minute stimuli inclinations. And you can see here, so the dashed line is for comparison. This is the maximal response for just one stimulus. So you see here, this is the maximal response. All of these dots represent the maximal response. So let's say the maximal angle, although it's a bit different here, after the second stimulus. So you see that for intervals of between 0 and, I don't know, half an hour, your response is indeed stronger than just one. So this is the addition. Between half an hour and an hour and a half about, your response is weaker than if it were just one stimulus. So this is a subtraction part. And here it's around 0. So after an hour and a half, you forget the older stimulus. Okay, so we're talking of a memory that goes back about an hour and a half in total. So this was very cool. We're very happy to see that plants have basic arithmetics. But we can ask, okay, apart from the fact that it's very cool, but why do they need it? Why is it useful in any way? Okay, and it turns out that other organisms use something called active sensing in order to enhance searching capabilities. So the importance of, if you're able to subtract two numbers, that's your way of comparing two numbers, because then if you subtract two numbers, if it's positive, it means that one is larger than the other. If it's negative, then it's smaller than the other. Otherwise, you have no way of telling which is which from a practical point of view. Okay, and if you can, so if you can subtract numbers and you can compare numbers, but over a specific point in time, then, for example, in bacteria, that's where they did something very similar to what we did here. In bacterial chemotaxis, they're too small to sense a gradient along their own body. So they sample their concentration here, then they go to some other place, they sample it again, and now they compare to what they had before. But it means that you need to be able to compare with that time scale. And now if it's okay, the gradient is going up, then you continue to go in that direction. If they see that it's not a good direction, then they will randomly choose a different direction. But not only bacteria do that, dogs do that. So here, it's hard to see, but this is a pheasant, and this is the smell trajectory, and this is a dog, and this is its head trajectory. So we all know the dogs do this, right, when they try to find a smell. And it's precisely that, because it's easier to sense a difference and say, okay, this is better this way, this way, this way, rather than always following the maximal signal. Bats do that. Practically any organism I know does that. So it's a really recurring theme. So why won't plants use that as well? And I think this is now conjecture. Okay, so hopefully I will be able to give you a better answer in a couple of years. But I think you see that if we look again at this movie of phototropism, see there are these, small circumutations. And it makes sense that plants would be able to, as long as you couple the same time scale of your memory with this time scale, then you're in business. It means that you can, when there's a weak gradient, you can sense the light here, sense the light here, and now determine which way to go. And some initial work that we're doing with different species seems to corroborate this, but with a very large chunk of salt. So we'll see. And I also say that this is also supported by some other recent findings in the lab, where we looked at sunflowers which are crowded together, so they shade each other, and then through shade avoidance, they basically, it was found out by a different group a while ago that they self-organize into a zigzag pattern. And when we analyzed the dynamics of how do they get to this zigzag pattern, we found these circumutations are actually, they actually facilitate this. So they're just the right amount so that it helps you find this zigzag. If it were less movement or more movement, you would be shading they would be shading each other more. So it makes sense in this broad picture. Okay, and with the last 2 minutes I have, I'll just say very briefly something about mechanics. So I talked about here how plants represent spatial information, how they integrate information over time, and how this could be useful for them. And another last thing is mechanics, right? In the end, a plant organ has also mechanical properties. Does it somehow use it somehow? So we want to understand this interplay between tropisms and passive mechanics. So we took a case study. I'll just say the story, the case study of these waving experiments that we all know, and want to say, okay, if we understand this because of the mechanical interaction with the substrate, we can say so that we understand something about this interplay. So we developed a nice numerical framework which allows to indeed incorporate mechanics with the tropism equations, and we're able to reproduce waving and coiling and predict the transitions from one to the other and have a lot of other information about it. That's it. So I just want to finish by reminding you the initial slide and hopefully convinced you that tropisms are interesting not only for their own sake, which is always true, but also they give us, they can open a window to something else. We can ask other questions through tropisms. And that's it. So thank you very much, and I'll be happy to get any questions.
Thank you very much, Yasmine, for a wonderful talk. So, questions?
Particularly liked the circummutation example you gave. And my question is, plants operate in populations. They don't operate as individuals. And so that behavior, did you observe that plants are bullies or they're well behaved?
You're talking about the sunflowers so I think I like to think of it as a dance party where you arrive to a party and here it's too crowded and there it's too sparse. So you just need to jiggle around and then everybody finds their own space. That's that. The jiggling around is the circummutations because it just allows them to explore their environment. That's what's good about it, because you might say that it doesn't make any sense to spend energy on moving in random directions, which are half of the time, by definition, not in the right direction, right? Because you're going to the wrong in terms of the light. But it does help them to explore their the area.
So it helps the population.
Yes, but this is also, it's not special to plants. This is something that's very well known in any collective system. If you think of flocks of birds or fish or whatever, there are always in the end 2 ingredients. You have local interactions, so I can only talk to my nearest neighbors, as we had here with mutual shading, but I always have noise, some sort of fluctuations, because if I have, and there's always a sweet spot, too little noise, and I get stuck in a bad solution. Too much noise, and I don't see the interactions anymore. And just in the middle allows me to, there's this explore, exploit kind of balance.
That's great. Thank you.
Thank you. Yeah.
Super talk. I'm really intrigued by the memory function work. And so we know, of course, a lot about auxin signaling and the molecular pathway. Have you been thinking or looking at studying the kinetics of what's known about negative feedback in that pathway and how that might relate to your memory function results?
So I'm not sure about the negative feedback, but we are looking at trying to find how this memory is encoded, right? Mm-hmm. And so my take on it is that it has to do with the stochasticity of transport processes. So I talk about auxin, it could be a million other things, but I take it as a kind of a flag bearer, like it moves information about a stimulus that occurred here to the rest of the organ, right? So in that mind, that's my representation. So if we think of the there are two light stimuli, okay? And this is the organ, and I'm at the back end where the auxins arrive. So I have two auxins that arrive at the same time. One is a fast auxin from the recent stimulus, and one is a slow auxin from the older stimulus, so to speak. But they arrive at the same time, just because, some are fast, some are slow. It's a random process. I mean, transport is you always have some kind of stochasticity involved. But this means that the two arrive at the same time, and I respond to both of them. So this is the summing, for example. And for the negative part, and I would be interested actually to ask a bit more about the negative feedback, because I'm not sure if I know what you mean, but there is this notion that at some concentration of auxin, there's a reorientation of auxin. So I don't know if that's what you meant, but that, from the modeling point of view, we have simulations, and it does reproduce what we see here. So now we're trying to connect it with real data, but
Thanks.
Okay. Yeah. There is here. Yeah.
Thank you. That was wonderful. My question is whether you can tell anything about how plants are dealing with noise in your models, because you could imagine that you could potentially have a continuous, maybe weaker light source, and then an intermittent, higher intensity light source, because the time-lapse movies you were showing were very noisy. And I'm wondering if you can sort of capture that, and it can tell you anything about the underlying biology, kinetics of the process.
You're asking how do they cope with external noise of the signals of the stimuli?
You could use your system, so continuous, perhaps a weaker light source, and then a higher intensity intermittent light source, similar to the sorts of things you were getting. Could you capture that in your models?
So already the memory function itself that I showed, actually, I'll just show one thing here. It turns out I sneaked in this dashed line, which is a fit to, this is the a forced oscillator, never mind. But the point is that it turns out that this is a classic from the field of signal processing, where people use filters to clean out data. So you have some noisy data or noisy stimuli, and now you have a filter that cleans it out so that you throw away stupid fluctuations, but you do identify trends. So it turns out that this is one of the classic filters. This is the bandpass filter. The positive part is a part which integrates, and this means that you remove all the small fluctuations. And the negative part, when I talk about subtraction, that means that you're basically looking at trends. It's like a derivative. So it turns out that plants have bandpass filters. And so what we want to do is indeed also to now look at it from the eyes of signal processing functions and quantify this. What does it mean? How can we, for different plants, are they better at this or better at this? What are the time scales? And precisely the sort of questions that you're asking. What type of, what we haven't done maybe and is important to show is to show which actual stimuli from around us are relevant for this.
Thank you.
Thanks.
Thank you very much. Very inspiring. I was just wondering the connection between mutations and really directional growth. If you also observe that if you don't give them a clear stimulus, for example, you look at a dark grown seedling, they do mutations very strongly. And then when you give them a target, so you start shining blue light, the mutation is almost gone. Then they shoot straight at it. What is this? I don't know if you observed this as well. We've seen this for phototropism, but we've also seen this for leaf movements where they do like this, and then you give a shade cue, and then they go very directly. Yeah. And what is this telling us about the underlying mechanics? Or
so I think it fits really nicely with the active sensing thing, because indeed we have experiments. Ronnie sweated on them a lot, where we have even homogeneous lighting, right? But let's say this two light setup with weak light, and then you increase and increase and increase, and you see that circumtations are much more pronounced the weaker the gradient is. So this makes sense because it means that if I don't have a clear signal, I need to use this active sensing. But if I have a clear signal, there's no need to waste my time on moving around in. So.
very much. Yasmine, we can go to the next speaker.